In 1869, half a century before quantum numbers were understood, the famous Russian chemist Dmitry Mendeleev produced the first accurate table of the elements. It was based on the number of protons and neutrons in the atom. He did not know that neutrons could be added or removed. In modern tables of the elements, the number of protons defines an atom's location in the table. Different versions of the table highlight different aspects of the atoms. This one highlights the number of electrons in each atomic shell around the nucleus. Here's a tabulation of how many of these elements are in living matter. The two questions we want to answer are: 1. How could these basic elements have combined to form the extremely complex molecules needed for life? And 2. How long would such a process take? In the How Small Is It video book, we covered the atom with its proton, neutron, and electron components, energy shells, and electron location probabilities via Schrödinger's equation. Shells contain multiple orbitals. An atomic orbital is defined as the region within an atom that encloses where the electron is likely to be found 95% of the time. Solving Schrödinger's equation using 4 quantum numbers and Pauli's exclusion principle yields multiple wave functions as solutions that define an electron's probability density cloud in an orbital. The electron's energy is quantized into electron shells designated by the letter n. It determines the distance the electron is from the nucleus. For each energy level n, the associated angular momentum is also quantized into electron subshells, designated by the letter L. For each quantized angular momentum subshell, the allowed orientations are quantized into orbitals and designated by the letters m sub L. And for each subshell, the electron's spin is quantized into 2 states, up and down, or plus and minus 1/2. Here are some images showing the shape and size of orbitals. N determines the size of the orbital. L determines the shape of the orbital. M sub L determines the orientation of the orbital. And spin limits the number of electrons in an orbital to just 2. The names of electron orbital angular momentums, S, P, D, and F, originate from early spectroscopy studies of atom spectral lines described as sharp, principal, diffuse, and fundamental. Electrons first fill the lowest energy orbitals available, gradually moving to higher energy levels as the lower ones are filled. When filling orbitals with the same energy level, electrons will first individually occupy each orbital before pairing up, because pairing takes more energy. Note that 4s is filled before 3d. This is because electrons in a subshell tend to repel each other more than electrons in different subshells. Here we have a subset of the periodic table that shows an electron distribution diagram for each element. These diagrams highlight the number of shells an atom has, the number of electrons in each shell, and the pairing of these electrons. The outermost shell is called the valence shell, and the unpaired electrons in the valence shell are called the valence electrons. Here are 4 key examples. It is the shapes and sizes of their valence electrons, along with their binding energies, that give atoms their chemical properties. Note that the diagrams show the paired electrons as close together. In actuality, because all electrons have a negative charge, and like charges repel, electrons in a pair will be as far apart as possible in their orbital. Molecules are formed when elements are combined. The atoms in molecules are held close together by attractions called chemical bonds. Forming a bond releases energy. We determine a bond's strength by measuring how much energy is required to break it. There are many kinds of chemical bonds, and each has its own range of strengths. We'll cover the ones that are critical for the formation and operation of living matter: covalent, hydrogen, Ionic, and van der Waals. Covalent bonds involve the sharing of a pair of electrons by two atoms. Here we have two hydrogen atoms approaching each other. If they get close enough for their electron shells to overlap, they will begin to share their electrons. Each hydrogen atom is now associated with two electrons in what amounts to a completed valence shell. The covalent bond has transformed the two hydrogen atoms into one hydrogen molecule. Its formula is written H2. This is a single bond because it is created by the sharing of a single electron. There are a number of ways to illustrate a molecule. One is called the structural formula. It focuses on the bond holding them together. Here, the single line indicates a single bond. Another, called the space-filling model, focuses on the shape of the combined atoms. And another, called the ball and stick model, is good for medium-sized molecules. The hydrogen molecule's bond strength is 4.52 electron volts. An electron volt is the energy acquired by a single electron when it is accelerated through a potential difference of 1 volt. Here's the oxygen molecule. It contains two oxygen atoms and is sharing two valence electrons and is therefore a double bond. Its bond strength of 5.13 electron volts is stronger than a single bond. Here we have one oxygen and two hydrogen atoms coming together to form a water molecule, H2O. They are joined by two single bonds. And here we have methane forming from a carbon atom and four hydrogen atoms. Of all elements, carbon is unparalleled in its ability to form the large, complex, and varied molecules needed for life. This is because it has four valence electrons. Carbon atoms represent a significant percentage of all living organisms. That's why any material containing carbon is called organic. Each of the four bonds has a strength of 4.3 electron volts for a total strength of 17.2 electron volts. The attraction of an atom for the electrons of a covalent bond is called electronegativity. The more electronegative an atom is, the more strongly it pulls shared electrons towards itself. In a covalent bond between two atoms with the same electronegativity, the electrons are shared equally. This is called a nonpolar covalent bond. Hydrogen and oxygen molecules are examples. When an atom is bonded to a more electronegative atom, the electrons are not shared equally. This type of bond is called a polar covalent bond. Water is an excellent example of this. In covalent bonds between oxygen and hydrogen, the electrons spend more time near the oxygen nucleus than they do near the hydrogen nucleuses. Because the electrons have a negative charge, this creates a region of partially negative charge, and it leaves the regions around the hydrogen atoms with a partially positive charge. Polar covalent bonds, in turn, enable additional molecular bonding called hydrogen bonds. We'll continue to use water as an example. The relatively positive charged hydrogen atoms in one water molecule are attracted to the relatively negative charged oxygen atoms in other water molecules. Water-hydrogen bonds are strong because oxygen has the largest electronegativity. Yet it's only about 1/20th as strong as a covalent bond. It's the hydrogen bonds between water molecules that have to be broken in order to turn liquid water into gas. We'll see later that it's hydrogen bonds that hold DNA strands together. Sometimes, two atoms are so unequal in their electronegativity that one strips a valence electron from the other. Sodium and chlorine are a good example of this. The transfer of an electron leaves one with a net negative charge and the other with a net positive charge. Atoms with one too many or one too few electrons are called ions. When two ions with opposite charges come close together, they form a bond called an ionic bond. Once the sodium atom loses the single electron in its third shell, it is left with only two shells. Its new valence shell is complete. It now has a net positive charge. The chlorine atom has completed its valence shell and now has a net negative charge. The ionic bond has transformed the two atoms into sodium chloride, salt. Van der Waals interactions are named after the Dutch physicist who developed the equations for the behavior of molecules and gases and liquids in 1873. We'll use two molecular oxygen molecules to illustrate how the force works. Most of the time, the negative charge around a molecule's nucleus is uniform. No force exists between the two molecules. But because electrons follow the laws of quantum mechanics, their positions can change over short periods of time. If they were to group on one side of the molecule, that side would appear negatively charged relative to the nearby molecule. And because like charges repel, it would tend to push the electrons in the other molecule back. This in turn creates a situation where the molecule on the left is relatively less negative, or equivalently, relatively more positive. And because opposite charges attract, we get a very weak and temporary attraction between the two molecules. This is the van der Waals force, and its strength increases as the number of electrons in the atoms, or the number of atoms in the molecules increases. These forces are ubiquitous in nature and influence the structure, stability, dynamics, and function of molecules throughout physics, chemistry, and biology. When comparing the strength of these bonds, we find that ionic bonds are the strongest, followed closely by covalent bonds. Hydrogen bonds are significantly weaker, and van der Waals bonds are the weakest. The making and breaking of a chemical bond, leading to changes in the composition of the matter, is called a chemical reaction. Here's an example. We have two hydrogen molecules and one oxygen molecule as the reactants. The chemical reaction breaks the covalent bonds of both the hydrogen and oxygen molecules and reorganizes the atoms. The product of the chemical reaction is two molecules of water. It's an exothermic reaction when energy is released. It's an endothermic reaction when energy is absorbed. Here we see that the energy absorbed to break the reactants' bonds is less than the amount of energy released when the product's bonds are formed. So this one is an exothermic chemical reaction. Here's a chemical reaction that ionizes water via a process called dissociation. It is very important in the chemistry of life. Occasionally, a hydrogen atom's proton participating in a hydrogen bond between two water molecules dissociates from one of the water molecules and associates with the other. Dissociation involves breaking the covalent bond, with the shared hydrogen electron remaining in the valence shell of the oxygen atom, and the proton forming a covalent bond with the other oxygen atom. The receiving molecule is called a hydronium ion. It has an extra positive charge having received the positively charged proton. It is referred to as a hydrogen ion. The other molecule is called a hydroxide ion. It has a negative charge. This reaction works the other way as well, where the hydronium ion loses the proton back to a hydroxide ion. These ions are very reactive. They participate in ionic, covalent, and hydrogen bonds with a wide variety of other molecules. Because it works both ways, an equilibrium is established in any given volume of water. There are approximately 33.4 trillion water molecules in 1 liter of pure water. Only one water molecule out of every 554 million is dissociated. Yet, this small percentage for ions gives us 60,000 trillion of them in a liter. In pure water, the concentration of hydroxide ions and hydronium ions are each 10 to the minus 7. Although the concentrations of hydroxide and hydronium are equal in pure water, the presence of other substances can change that. A substance that increases the number of hydrogen ions in a solution is called an acid. Hydrochloric acid is a good example. It loses its hydrogen atom to a water molecule, adding an additional hydrogen ion to the solution. A substance that decreases the number of hydrogen ions in a solution is called a base. Ammonia is a good example. it takes a hydrogen ion out of the solution. The relative abundance of hydroxide ions defines pH values and has a dramatic influence on how molecules behave in a watery solution, including the inside of a living cell. In fact, the internal pH of most living cells must remain close to 7.2, including the synthetic living cell, JCVI, synthesis 3A. We'll study this cell a bit later. The shape of a molecule has a great deal to do with its function. For living matter, carbon is the key element for a wide variety of shapes. Here's the methane molecule we saw earlier. The red lines outline its shape. This one has a three-dimensional tetrahedron shape. This will be the shape whenever a carbon atom forms a molecule using its 4 valence electrons to combine with 4 other atoms, including carbon. Here's ethane with 2 tetrahedrons pointing at each other, and here's ethene. It shows that when 2 carbon atoms are joined by a double bond, all other attached atoms will be in the same plane. The molecule is flat. These are all hydrocarbons, organic molecules that contain only carbon and hydrogen. Carbon chains form the skeleton of most organic molecules. These skeletons can vary in length. They can branch. The locations of single or double bonded can vary, and they can even form rings. Here are the 7 functional groups that are the most important in the chemistry of life. They attach to carbon skeletons to produce organic molecules. The number and arrangement of these groups give each molecule its unique properties. Here are a few relatively small molecules that use these chemical groups. The top half are acids found in proteins: asparagine, glycine, tyrosine, and alanine. The bottom half are bases that are found in DNA: guanine, adenine, cytosine, and thymine. The larger molecules that life depends on are built from combining smaller molecules like these acids and bases. There are 4 classes of large biologically important molecules: lipids, carbohydrates, proteins, and nucleic acids. The last 3 of these can grow into large, complex macromolecules, so we'll focus on them. Macromolecules are built from polymers that are made up of large numbers of smaller molecules, called monomers, that are bound together by covalent bonds. The diversity of life, with its trillions of different organisms, are made of polymers that are constructed from just 50 monomers. We'll take a look at carbohydrates, proteins, and nucleic acids to see how this works. Carbohydrates have monomers like glucose and fructose. They can be joined by covalent bonds that form the polymer sucrose, table sugar. Carbohydrate macromolecules, like starch and polysaccharides, form by combining a few hundred to a few thousand of these polymers. Proteins account for over 50% of the dry mass of living cells. Protein monomers are built from just 20 amino acids. Most of them have a tetrahedral arrangement of the atoms. The monomers combine into polymers called polypeptides via covalent and ionic bonds. A protein macromolecule is built from these polypeptides. They are the most structurally sophisticated molecules known, with each type of protein having a unique 3-dimensional shape that determines its function. Van der Waals interactions play a crucial role in stabilizing the protein structure. There are 2 forms of nucleic acid: deoxyribose nucleic acid, DNA, and ribose nucleic acid, RNA. We'll focus on DNA in order to understand the complexity of its structure. A DNA monomer is called a nucleotide. It has 3 parts. The first is one of 4 nitrogen bases: adenine, A, thymine, T, cytosine, C, and guanine, G. We'll start with cytosine. The second is a deoxyribose sugar, S, joined to the base by a covalent bond. The third is a phosphate group P joined to the deoxyribose by a covalent bond. This is a DNA nucleotide. C's and G's bind together and A's and T's bind together. They connect via hydrogen bonds to form base pairs. These base pairs are bound to other pairs by a covalent bond between the phosphate groups and other deoxyribose sugars. Repeating the process gives us the DNA molecule. This is its structure diagram. Here's a ball and stick view. The bases come in two categories: the pyrimidines with a single ring and the purines with two rings. These bases are used by all living things on Earth. Here we have a random collision of a cytosine molecule with a deoxyribose nucleic acid. In the chemical reaction, the weaker covalent bonds between nitrogen and hydrogen in the cytosine and between carbon and oxygen in the deoxyribose are broken. Stronger covalent bonds are formed between the hydrogen and oxygen atoms forming water, and between the nitrogen and the carbon atoms. Next, we have the bonding of the new larger molecule, called a nucleoside, with a phosphate group. Again, old bonds are broken and new bonds are formed. This is a DNA monomer, a nucleotide, a building block of DNA. Now we move in a guanine monomer and join it with the cytosine monomer. Together they form a base pair, held together by hydrogen bonds. This is a full DNA horizontal component. This GC example contains 72 atoms. As we saw earlier, the electron location probability distribution around each atom join to form a complex cloud around DNA monomer base pairs. These base pairs are stacked one on top of another, joined by covalent bonds between their end phosphate and sugar components. But as two base pairs randomly close in on one another, direct alignment creates electron-electron repulsion and Pauli exclusion principal problems that prevent any bonding between the two. But with a rotation of 36 degrees, these conflicts disappear and the bond is completed. Careful measurements found that they are only 0.34 nanometers apart. That's close enough for van der Waals interactions to help hold them together. Building on each other, these complementary base pairs create the full DNA molecule. The 36-degree twist per layer would produce a 360-degree rotation every 10 layers, a helix. A single human DNA molecule contains approximately 3 billion base pairs. That's around 216 billion atoms. There are as few as 20,000 base pairs in a virus DNA. But viruses need a cell in order to reproduce. So the first life could not have been a virus. The simplest single-celled organisms are bacteria. Here's Mycoplasma mycoides. It has 1.08 million base pairs and contains 901 genes. We'll take a closer look at this bacteria to see how it was used to create the simplest possible living cell. In 2010, the J. Craig Venter Institute, JCVI for short, announced that they had created a synthetic living cell. They fabricated the DNA in a lab and used it to replace the natural DNA in an M. mycoides cell. They knew that they had succeeded when the cell began to reproduce with its new genome. They named their synthetic life form JVCI, SYN 1.0. 6 years later, the team announced that they had created a synthetic minimal living cell containing only the genes necessary to sustain life in its simplest form. It had 531,560 base pairs and just 473 genes. They named it JVCI SYN 3A. Here's an illustration of SYN 3A's cross-section as it is reproducing. It shows all the key macromolecules. For acids, we have DNA in bright yellow and RNA in shades of magenta. For carbohydrates, we have the membrane with some complex carbohydrates like polysaccharides in green. For proteins, we have those associated with DNA in tan and orange, synthesis factors in purple, enzymes, which are proteins, in shades of blue, and metabolic enzymes in shades of turquoise. For life to have formed on Earth, the starting mix of available atoms would have to combine into simple molecules. Then these simple molecules would have to combine into more complex molecules. Then these smaller complex molecules would have to combine into even more complex monomer molecules. Then these molecules would have to combine into the macromolecules of life. What processes could have enabled these transformations? And for our purposes in this How Old Is It video book, how long would those processes have taken? In the next segment, we'll cover the first experiment that attempted to answer this question.